unlocking the secrets of genetic adaptability. In the face of climate change, the need for resilient crops has never been more pressing. Researchers are racing against time to develop plant varieties that can withstand the challenges posed by our rapidly changing environment. But here's where it gets controversial. Can we truly harness the power of genetic switches to ensure food security for future generations? The importance of regulatory DNA. A groundbreaking study led by an international team, including experts Thomas Hartwig and Julia Engelhorn from the Max Planck Institute for Plant Breeding Research and Heinrich Heine University Düsseldorf, has unveiled a novel method for mapping regulatory DNA segments. These segments, often referred to as switches, play a pivotal role in controlling how and when genes are expressed, which is essential for developing plant varieties that can thrive in challenging climates. Understanding genomic variation. In their research, the team analyzed 25 diverse maize hybrids and discovered over 200,000 regulatory regions that significantly influence vital traits such as plant height, leaf shape, and resilience to drought and diseases. Astonishingly, even though these regulatory switches constitute less than 1% of the entire genome, variations in these areas can account for a considerable portion of heritable differences in plant traits, sometimes more than half. This revelation opens up exciting new possibilities for plant breeders, allowing them to focus on these regulatory switches to expedite the creation of climate-resilient crops. Thomas Hartwig emphasizes the significance of this research, stating, "By understanding how these regulatory switches function, we gain powerful new tools to enhance both crop resilience and yield, paving the way for smarter agricultural practices in the future." Targeted breeding for drought resilience. The research team specifically targeted drought stress, identifying over 3,500 individual regulatory switches and their corresponding genes that respond to water scarcity. This precise mapping allows for targeted manipulation of these switches, enabling the development of plants that can better withstand drought conditions. Julia Engelhorn highlights the innovative nature of their approach. Our hybrid-based assay facilitates a direct comparison of maternal and paternal regulatory alleles within a single experiment. We are now providing the research community with a valuable resource of over 3,500 drought-linked regulatory sites, which opens up new avenues for fine-tuning gene expression to enhance plant robustness. A new perspective on the non-coding genome. Co-author Samantha Snodgrass from the University of California, Davis, points out a significant shift in our understanding of the genome. Despite decades of groundbreaking research on genome evolution, much of the non-coding genome has remained a mystery. This exciting new method sheds light on these areas, offering breeders and biologists precise targets in regions that were previously overlooked. As we stand on the brink of a new era in agricultural science, the implications of this research are profound. Can we truly leverage these genetic switches to create crops that not only survive but thrive in the face of climate adversity? What are your thoughts on the potential of this groundbreaking approach? Join the conversation in the comments below.